when you have multiple sets of chromosomes or copies of the information, we refer to this number or the number of copies you have as the ploidy. For example, haploid cells, represented by an N, have one copy of each chromosome. Gametes are haploid sex cells, that is, cells specialized for reproduction that have one copy of each chromosome. Diploid cells, on the other hand, have two sets of each chromosome, one from mom and one from dad. Now, all the cells you find in your body, aside from the sperm or the egg, which are the human gametes, are diploid, meaning they have two sets of chromosomes, one from mom, one from dad. The technical term for these body cells, right, so your skin, your blood, your muscle tissue, right, these are called somatic cells. So ploidy is a little confusing, what haploid means, what diploid means, but I think I have an analogy that works pretty well, and certainly one that I'm familiar with, and I think maybe many of you are going to be familiar with as well. Harry Potter. Okay? So, let's make the analogy that the Harry Potter series are a set of chromosomes, with each book representing a single chromosome. So there are 7 Harry Potter books: The Sorcerer's Stone, The Chamber of Secrets, The Prisoner of Azkaban, The Goblet of Fire, Order of Phoenix, The Half-Blood Prince, The Deathly Hallows. Now, assuming you have only one set of these books, or chromosomes, what would your ploidy be? Well, if you said haploid, you would be correct. So this is how you'd write haploid, right, with 7 chromosomes. You have N to designate haploid, you have one set, and then there's 7 chromosomes in total. Okay, now let's assume you also happen to have the British edition of the Harry Potter series. It's the same 7 books, the same major storyline in each book, but now we might have some kind of British slang thrown in there. So any guesses as to what the ploidy of the ploidy of our set of Harry Potter books? What is that ploidy and how would we write it down? Any guesses? It would be diploid, since we have 2 versions of each book, right? So you have a British version and an American version. 2 versions, diploid. So you write that as 2N, and we have a total of 14 books, or 14 chromosomes. So it would be 2N equals 14. Okay? All right, now let's get back to our stick diagrams of a chromosome so we can kind of quickly talk about the structure. Okay, so along each chromosome, there are particular regions that code for specific genes or instructions for a protein. For example, earlobe shape or eye color. Now, the same region on each chromosome codes for the same basic trait, regardless if it's on the chromosome from your mom or your dad. The specifics of that trait may vary, right? So, like your mom might have green eyes while your dad has blue eyes. But that eye color gene is found at the same location. Now, getting back to our Harry Potter analogy, you can almost think of them like book chapters. The first book in the British version and American version of Harry Potter are basically the same storyline, but you might have some different wordage here and there, right? Some different slang thrown around. Now, things get a little tricky during the cell cycle. So just before a cell replicates through cellular division, one of the steps in this process is making a copy of the DNA to pass over to the new cell. So before mitosis, the actual process of cellular division, a copy of each chromosome is made directly adjacent to the original, forming something called sister chromatids. Now in this case, ploidy does not change, right? So even when you have 2 copies of the same chromosome next to each other, called sister chromatids, your ploidy hasn't changed. Why? Well, there's no new information. There's no new version of the chromosome. There's not like you added a third parent into the mix. You just have the existing ones. Getting back to Harry Potter, maybe this might help. It would be like you take your two versions of the series, the American and the British version, and you photocopy them. Our ploidy in this case is still 2n equals 14 because we haven't introduced any new versions or any new text or any new slang, right? We just have copies of the same existing versions. Now, if we were add what's the sequel that can't think of? There was like a play originally, turned it into a book. It was like, okay, I can't remember. But if you were to add that book at the end here, then it would be 2n equals 16, right? You'd have a British version and an American version of that sequel. Anyway, let's stick with our 2n equals 14, the original series. The point I'm making here is photocopying your books versus chromosome duplication and ploidy, same principles apply. Just because you have copies doesn't mean you've changed your ploidy. You need different versions. Okay, so now we're on to the cell cycle itself. Just a brief introduction, then we'll take a break, and we'll come back for lecture 10.2. Now, first off, all cells must divide, either for reproduction or growth, right? The overall goal of the cell cycle is to make identical copies of the original cell, in pretty much every way, including and especially ploidy. This means that the ploidy that you start with during the cell cycle is the ploidy that you end with. Or, if you're starting as a haploid cell, you end as a haploid cell. If you start as a diploid cell, you end as a diploid cell. Start as a 100 end cell, end as a 100 end cell. I'm not really sure how you'd say that though. Centiploid? Anyway. So, we keep saying that all cells need to divide, and we keep talking about specifically eukaryotic cell division and mitosis. How do prokaryotes do it? Also, quick question, maybe a bit of a throwback. Which two domains are included in prokaryotes? You guys remember? Archaea and bacteria, right? So prokaryotes do something called binary fission. So it's a much simpler process where they basically just double in size by creating more phospholipids in their plasma membrane, making a copy of their chromosome, and then splitting into two new cells. It's much simpler than eukaryotic cellular division. Why? Well, they have a much simpler internal structure, right? So they only have one circular chromosome. That's pretty straightforward. You don't have all these chromatin hanging around. You also don't have to worry about dealing with all those organelles like the nucleus with that super complicated membrane and the Golgi and all that other stuff. You don't have to deal with any of that. So, interesting point. If they only have one circular chromosome, what does that mean for their ploidy? Any guesses? Right, they're haploid, okay? So they only have one copy, it's one circular chromosome, most of them are haploid. So at the end of binary fission, a prokaryotic cell goes from haploid to haploid, or N to N. Straightforward. Eukaryotes, on the other hand, are a little bit more complicated. They go through a cycle consisting of two major phases. Interphase, in gray here, where the cellular growth and DNA replication happens, and the mitotic phase, where the actual process of eukaryotic cellular division occurs, also known as mitosis and cytokinesis. Now, we're going to talk a lot more in detail about that next lecture, in lecture 10.2. There you go, yeah, we'll talk about interphase and M phase, or mitotic phase. Before that though, let's look at lecture question 10.1, compare and contrast which of the following are true about chromosomes. So do your best, and talk to you next lecture. Bye.